All right. Well, I hope all 70 of us are hunkered down in a nice cool basement, or glory be, actually have air conditioning. Because it is a hot day, and I was thinking about the lichens also hunkered down on their trees, probably tinder dry. But trust me, they're handling this desiccation quite well. So why? What an annoying question, right? It's a question that you get asked if you're a parent a lot. It's a question that I get asked as a taxonomist a lot. Why lichens of all things? Why should I care about lichens if I'm not a lichenologist? Why biodiversity? Why do you work for the ABMI? So I hope today we'll be able to answer some of those questions, and maybe at the end of the program it'll be more like why not just dive deep into this amazing group of organisms, communities of organisms. So Brett did a great little introduction to ABMI, so I will keep this quick. But we know, I mean, if you're at this seminar, you appreciate biodiversity, and biodiversity is just simply all of that amazing diversity of landscapes, species, and individuals that are surrounding us, that are feeding us, that are providing us ecosystem services, that are filling up our Snapchats and our Instagrams with the amazing Albertan landscapes. I hope one thing you noticed when in that little montage was that a lot of the biodiversity we see is vegetation, it's flora, it's plants. And if we went to those same landscapes and we looked with a slightly different lens, if we peered down in between the dried grasses, and if we looked up at the trees, we would see a whole nother layer of biodiversity. And lichens are a part of that layer. And today I'm really hoping to kind of help you take your blinkers off and learn to appreciate and marvel at this amazing group of organisms. And Charles Darwin, I think he said it best. So a traveler should be a botanist, for in all views, plants form the chief embellishment. And plants are a critical part of Alberta's biodiversity, and lichens are a portion of that. So there's a lot of kind of touchy-feely reasons, ecological and functional reasons to care about biodiversity, and that has led to legislative and policy reasons to also care. So even if you're not touchy-feely about this stuff, there's good legal reasons to care about the other species that we coexist with. As Brett mentioned, ABMI is a province-wide systematic sampling program. It's one of the unique attributes about it is that we go to different sites across the province, and they're not chosen to land in a certain habitat type. Basically, there's our core program is 1,656 sites across the province, each of these little pale dots. And at each of them, we get as close as we can with landowner permissions, and we do biodiversity surveys there. So it's meant to be a long-term systematic view of the species and the habitats that occur there. We do have both terrestrial and wetland components. We look at habitats and species in the field. And we, of course, look at landscapes using remote sensing province-wide. And we use standardized methods. So I'll be talking about the bryophyte and the lichen protocol today. ABMI is pretty unique, but there are a few programs for those of you who may not be from Canada or who want a more international perspective. There are a few other programs around the world that share some of our philosophies. And those philosophies are we want to be broad scale, we want to be long term, and we want to be multi-taxon, because biodiversity, it's not just birds, it's not just beavers, it's not just trees. It's about polling and looking at species with different traits and different habitat requirements and trying to come to some kind of consensus on how they're faring with how the world is doing. So, of course, biodiversity is one of these also incredibly frustrating concepts because you just cannot encapsulate it in a way that satisfies anyone. It's either too broad or it's too narrow. So you have to have a pragmatic approach to it at some point. And ABMI, over its history, it attempted quite a few different taxa, including things like bracket fungi, fish, algae. And at the end of the day, well, right now, where we are in our history, we have 8 core taxa that we monitor. And this is kind of known as a bet hedging strategy, or think of it as polling 101. Basically, these are the taxa that were logistically feasible with the cost constraints and the time constraints. And Alberta has some challenging time constraints given we have a relatively short field season. And these taxa also encompass a diversity of life histories, traits, habitat requirements, and different societal values to people. Sometimes I hear, ABMI really, what don't you monitor? So I just wanted to make the point that there's a lot that we don't monitor. And I'm afraid, if you're an invertebrate zoologist in the audience, you are a tricky taxon, and we have not managed to do much in the way of like Hymenoptera or flies, but we have some fantastic aquatic invertebrate and soil or batted programs I hope you'll come and hear about in the future. But the take home message here is depending on what estimate you look at, Alberta has between 36 and 80,000 species, only about 12,000 of which we know and can actually give like some kind of conservation rank to. And even optimistically, we're only getting data through the ABMI, about 5 to 10% of those, and enough data. So these are the common species to construct habitat models for 2 to 5%. It's still an incredible achievement, but it's certainly not monitoring everything. So that's enough about ABMI. Let's ask the question, why lichens? Why did they make the cut? Lichen 101. So lichen, basically what is a lichen? Um, if you can imagine, over the last 500 million years, various fungi, and a lot of them were sac-forming fungi, um, basically formed partnerships with different cyanobacteria or things like nostoc or green alga. And they formed this amazing partnership that we call a lichen. So a lichen is not one thing, and lichens are not even an evolutionary group. They're an ecological group. It's basically like a diet choice. These are fungi that have learned to coexist with algae, and the algae kind of bring home the bacon. They photosynthesize and make sugars, and the fungi, for the most part, are providing structure and sometimes chemical protection. So they're like the carpenter of this relationship. And together they're forming 17 to 20,000 different unique communities that we know as lichen. So if we take a slice through that leafy lichen we were just looking at, and we kind of cartoonize it, we start to appreciate the fact that lichens are not one thing. And so when you monitor a lichen, at a minimum you're monitoring two different organisms, your fungus and your alga. A lot of lichens also, as we're learning, have basidiomycete yeasts in their cortex. Other lichens are actually three-partnered lichens. They have green algae and they have cyanobacterial algae. They have amazing microbiomes, other fungi and viruses and bacteria that are living within their bodies. And they house invertebrates. So if you've ever looked at lichens under the microscope, you too may have been surprised when a gorgeous little predatory mite popped its head out from under a lobe. So there is so much nested diversity in lichens, and if we take away the cartoon and just appreciate the beauty of the lichen in a scanning electron microscope image, we can see at the top here, this is where the cyanobacteria are living in this particular leafy lichen. And in this relationship, the fungus is able to kind of upregulate that amazing nitrogen fixing capacity because, of course, cyanobacteria are one of those organisms that can take atmospheric nitrogen and fix it into a form that other organisms can use, which is just a critical ecosystem function. And then the next layer here would be our green alga. And then we have these beautiful threads of fungal hyphae. So why lichens? We know a little bit about what they are now. Why do we want to monitor them? Well, they are diverse. We we can just admire some of the forms and the gorgeous functions. They're coating trees, they're coating soils, they're coating logs. Some of them are as tiny as under a millimeter. Some of them are as big as the palm of your hand. So there's an amazing diversity of lichens, and they occur basically in every terrestrial ecosystem in the province. They are sensitive. So a lichen basically lives, I like to think of it kind of in the cracks, in the dawn and the dusk of the day, on the dust and the mist that falls on them over their lifetime. And they sit there hunkered down, until they get some moisture so that they can photosynthesize and come back to life. And when that moisture arrives in them, it takes any dust that's settled on their bodies and it kind of transports it into their bodies, so they get the minerals and the nutrients that they need that they can't make. And they don't have roots like plants do. They're long lived. Some of these lichens that we're looking at might be 20 or 30 years old. And so all of these traits combine to make kind of a like sponge-like community that's sitting there and absorbing whatever we are throwing at it or what the ecosystem is throwing at it. And some of them can deal with it very well. They're resilient. They're the lichens that we see in our boulevard trees. And others are going to just be incredibly sensitive and they're going to drop off very quickly. So that sensitivity and that range of sensitivities is also what makes lichens a great eco monitor. They're also widely distributed. You don't want indicators that are very narrowly distributed when you're looking at a broad scale monitoring program. So as I mentioned, we get lichens in boreal forests, we get lichens in rocks in the grasslands, and we get lichens in trees in the foothills and the Rocky Mountains. Sometimes they're the same, and some of these lichens, you could almost draw our Alberta natural region boundaries with their distribution. They're so tied to the climate and the biogeochemistry of our ecosystems. The last kind of important characteristic for an indicator is that they're seasonally stable. So as I mentioned, these things live for a long time. Here we have a bunch of beautiful lichens in the winter time. If you're studying lichens that are not covered by snow, you can study them year round. So that is a really helpful logistical trait. It allows our technicians to go out in May, June, or July to assess lichen diversity. um, they're colorful, so they're visible and detectable. And lichens are also supported by more than 150 years of science, which is kind of mind blowing. Um, a lot of this science dates right back to some observations of gardens in Paris and how some lichens were disappearing as air pollution started to increase. And conversely, some of those same European cities are noticing lichens come back as we start to, decreasing the levels of things like sulfuric dioxide in our ecosystems. So they're fantastic indicators of air quality, habitat quality, and more we're using lichens as indicators of climate because again, they're so driven by their moisture requirements. So just a little case study, we've used that sensitivity to construct an index of atmospheric purity, which is kind of a funny term, but basically it's a way of mapping lichen diversity across the city. And this was a project to test, well, basically to ask lichens what was happening in Edmonton. And so a group of students from the University of Alberta taking a Lichens of Alberta class, they went to trees on a 1.5 by 1.5 kilometer grid right across the city. And they counted up how many lichens they found and how abundant they were. And then PhD candidate U of A Christopher Schulze did this analysis for us. So the areas with the red are showing lower lichen diversity and cover, and the areas with green are showing higher diversity and cover. And if you live in Edmonton, you probably have some hypotheses about why these patterns are existing the way they are. We have some old city airports, and we have some pretty hot urban island right in the downtown there. Conversely, we have some riparians and river valleys down here in the greener sections. So lichens are responding to the climates and the air quality um, that more and more we are influencing. So why would you want to use lichens when, Edmonton does have air quality stations? Because we can't afford to have the number of air quality stations that lichens provide us for free. So here's a great example of an ecosystem service that they can do. They occur on almost every boulevard tree. Very few trees have no lichens whatsoever. They're inexpensive to survey. And you can go out and look in your neighborhood and see what the lichens are telling you about your climate and air quality. So that leads us to ecosystem function. And here's a list of just some of the functions that lichens contribute to. An ecosystem function, again, it's one of those words like biodiversity. It can be hard to measure. kind of maybe easy to conceptualize, but basically these are things that the other 99.999% of species on the planet um, do as a result of their, their coexistence, that otherwise we might have to pay for. So things like fixing that nitrogen, so we have nitrogen available to us, fixing carbon to help us with climate change issues, stabilizing soil or keeping water in the soil, which is critically important right now, as you can imagine, or even providing food to some of our favorite charismatic megafauna. So some of the lichens that provide the biggest bang for your buck are, again, these leafy lichens we call peltigera. And this is just a little scan of some of the diversity we can find right in our Edmonton River Valley. This is just below the University of Alberta. And it turns out Alberta is incredibly diverse in these lichens, which we call cyanolicans because they have cyanobacteria as their algal partner. We did a survey with Duke University in just a 20 by 20 meter site. And I think it took two of us over 5 hours and we came away with 150 different collections. So I think it's underappreciated how diverse, how abundant, and thus how important these organisms are. There was a nice little case study published in Nature Geoscience. So if you're a kind of person that likes to have some numbers, they estimated based on the science of the time that biocrusts, and biocrusts are just this thin skim of life on the planet. They're composed of lichens and mosses and algae and fungi and bacteria. So as a composite group, these guys take up over 3.9 billion tons of carbon per year, and they account for about half of the biological nitrogen fixation on land. So they're doing us a pretty darn awesome service there. So hopefully you're a little bit more convinced now than you were 15 minutes ago about why we monitor lichen. I want to take you through quickly on how we monitor them because this is a question we get quite a bit, and they are a tricky group. Um, I've spent over 10 years, maybe it's now 15 years, I've lost track, trying to develop some expertise in this group, and I am still learning so much all the time. So how do you take a technician and train them in a few days to monitor this incredible diversity? Well, we do it based on an approach that is used by the Forest Health Surveys in the United States, developed by Bruce McCune, and also by kind of what is called a floristic method that was developed, well, maybe best published by Stephen Newmaster at the University of Guelph. Um, so it's basically saying that we know that a lot of lichen diversity is driven by tiny little niches through the landscape. And if we can go to those niches and collect from those niches, we will capture a lot of the diversity present. So when we have a habitat like that, and this gorgeous little lichen, this is what's sometimes called an old man's beard or Usnea hirta, bristly beard lichen. How do we go from the forest to the sample to the dot plot map to the habitat model? Well, it kind of starts by going to the site. So here we have a map of Alberta showing the 1,656 sites. And each of those sites might look big, but each terrestrial site is about one hectare in size. So there's some bird and mammal surveys that are done at a larger scale, but the flora is monitored at that 1 hectare scale. So when we get into that 1 hectare, what do we do to assess these mosses and lichens? Well, we break those microhabitats, or there's mini niches, into different strata. So logs and stumps, rocks and cliffs, trees, wetlands, and upland soils. And we do the three that are the most diverse for lichens in a plot kind of survey, and we do the two that are more on your hands and knees, nose down, bum up type surveys on a belt transect. And we do this 4 different times because we don't want the technicians to have to worry about assessing like a relative abundance metric. So we get that from the actual repeated surveys in the field. So as I talked about microhabitats, what exactly does that mean? Well, let's just scan through them pretty quickly. There's logs, and of course, logs of different decay classes and logs that arose from different tree species, rocks of different types, granitic versus sandstone. different aspects of the rock. Trees are an amazing diversity enhancer for lichens. So we teach the technicians to survey everything from the tips of the twigs to the roots that are exposed at the base of the tree. And we include in here human structures because remember, these are standardized surveys that we're doing right across the province, regardless of what type of forest or non-forest ecosystem we land in. So we look for these strata at every site. If they're not there, we mark them as missing. And if they appear in a repeated survey, that tells us something additional about that site. Upland soils, including agriculture, can be rich, as can wetlands, even focusing in on the really cryptic little black jelly lichens that occur sometimes on moist soil. So by the end of the training, when a technician looks at a forest like this, they're starting to mentally break it down into those strata and those little microhabitats. And they're going through that systematically and sampling even within diversity across those microhabitats. The training is iterative. It takes time. It starts in the classroom, looking at morphological diversity and starting to understand what a microhabitat is, doing group surveys that the instructors come along and check their results and point out anything they missed, doing practice plots or mock days where they actually bring their collections in and again are assessed. We spend quite a bit of time in the field doing this kind of iterative training with them, bring things back to the classroom. So again, we can point out what they might be missing and compare their results across practice plots. So it's a fair bit of work, but I think the technicians actually quite enjoy it. Sometimes they, I mean, our technicians are bio nerds, and so they really appreciate having their eyes open to these new taxa. So they're out there working hard, collecting samples for us all summer long, sending them back to us in what we call the giant Santa sack. That looks a little bit scary, but in most of these ecosystems, if you're getting a bag this size, there's probably, I couldn't even care to guess how many hundreds times more volumes of lichen present at the site. Those collections get dried, and here are this year's collections, which have just made it into the freezer to stop any activity of those pesky little mites that like to munch on the samples. After the samples are in, we spend time in the lab. The students learn to identify the most common ones, and we've shown from the pilot period that with training, these guys can be, 90% plus accurate in identifying a pretty large suite of species. And at the end of the day, the end of the summer, we have boxes of specimens that my team, my wonderful, hardworking team, then spends the next year identifying. And in total, we probably process between 15 to 25,000 specimens annually. So the rest of that year, I'm not going to do its service, but it's spent looking through the microscope. I am a microscope wonk for most of the day, for most of the winter. We do sneak in some field work so that we can explore maybe a species that we don't understand. We need to go to the source to collect a bigger sample. It's like any science, it's constantly evolving, taxonomy or the science of naming things. So we have to stay abreast of the molecular updates that are happening constantly. We might be using advanced imaging like scanning electron microscopy to understand certain species or visiting herbaria. And of course, we get to play with chemistry. This is maybe a unique thing if you're a botanist. This is one of the few fields, unless you're a mycologist, where you're spending time with the white lab coat on, working with solvents. And that's because lichens are deceptively simple, but they produce a bounty of what we call secondary metabolites, or chemicals that they don't really need for reproduction or everyday growth or living, but which are obviously critically important to their partnership. So a lot of these chemicals are things like sunscreens or antivirals, funnily enough, or even antifungals. They might be a predator deterrent in that they taste bad. So here's a little Alberta lichen. It's actually named after lichen. So it's named after Alberta, pardon me. It's Melanolixia albertana. And it has in its middle a chemical that reacts pink when you add a little bit of bleach. So when in doubt, you can pull out a toothpick and a little bit of bleach and touch it to your Alberta lichen to check its identity. We also spend a lot of time working with collaborators right around the world to make sure that our taxonomy is the very best it can be. So we are really, I think, privileged to have collaborators at Kew Gardens in the United Kingdom, all across North America, of course, Duke University for the Peltigeras, just an amazing group of people that are helping us do our jobs to the best of our ability. So at the end of the day, what are some of the highlights that all of this work is leading to? And how are we actually using these data to track biodiversity and conserve Alberta's biodiversity? Well, one of the messages that maybe you're not surprised about, I think most of the time if you dig into biodiversity, you're going to find some new things. Alberta, you're richer than you think. So there were about 900 species on our provincial list when we started the program, and we probably upped that for another 100 or so. And some of them are new to science and are still being described. Some of them are new to Canada, and a lot of them are just new records for Alberta. So things that we thought stopped out east, but are actually making their way all the way into the prairies of Alberta, or conversely, peeking their heads in over the mountains from BC. This is a pretty sample of the Lippy frost lichen, and it's called the Lippy frost lichen because it has these little powdery granules on the undersides of its lobes. And it's a new species to science, and the what we call the type specimen, or the specimen that all lichenologists will look at to understand this new species concept, is from the Hinton area. We also have a new little tiny lichen to science. This thing grows on dead rose, which is totally unique within the group. But when you look at it up close with a scanning electron microscope, doesn't it look cool? Kind of looks like an alien to me. And those, what looks like maybe little spaghetti noodles or rice grains poking out the top, those are the sexual spores. So what a fun job I have. The mini lichens. So we monitor these things called stubble lichens or whisker lichens. They tend to be under a millimeter tall. They've also been used extensively across North America and parts of Europe as habitat quality indicators. We've doubled this flora. So I believe there are around 25 species known when we started the program, and we're up at over 50 now. If we think about where those species are occurring, you probably won't be surprised to hear that most of them are in the forested regions. So just because trees are such an enhancer of lichen diversity, they're basically like hangers just waiting for the lichens to grow and drape and attach themselves to. If we look at the boreal, and we just do like a really simple summary to show you how diverse these species are. And these are from ABMI data, so they're of course affected by our choice of methodology. But if we compare the number of vascular plants at an ABMI site versus the number of larger lichens that we survey and bryophytes, the maximums are quite close to each other. And the averages might surprise you. We get up to 33 species on average, but right up to 73 at the maximum in a boreal site. And boreal sites are, a lot of their diversity is in the trees. They don't have the same rock diversity as you get in the shield in the mountains. So we can get well over 80 species in the rock-eared tree habitats. But lichens are very vulnerable to human footprint. And then human footprint is kind of an ABMI term, but it basically means physical alteration of the habitat. So it's something like forestry or agriculture, oil and gas, where we actually change or remove elements of the environment. And when we do that, when we look at sites that have had 50% of their area altered by human footprint, we can see that lichens decline more than the other floristic groups. Vascular plants will actually increase a little bit because you start to get the weedy species. Well, it turns out there are almost no weedy species of lichens. And to my knowledge, there's only one, well, this is kind of new news, one kind of invasive species of lichen present in Alberta. And it's not going to be very invasive for a very long time, but it's living in Edmonton. So if you want to ask me about that after, you can. They decline quite precipitously as we add human footprint to a site. And this is even more pronounced when we look at a prairie ecosystem, which is less diverse to start with. But as soon as you start to disturb those soils or those biocrusts, the lichens start to drop off. So all of that work goes towards what I've sometimes heard called the dot maps, and the dot maps are they're deceptively simple, but they're telling us so much information and they're the basis of all of the other modeling that happens. So we can use that dot map and we can put in other predictors like natural regions and types of human footprint and climate. And we can start to estimate what we think the abundance of individual lichens were across Alberta over time. So if we take away human footprint, that would be our reference. If we add back that human footprint, this is how we predict the abundance has changed. And I should point out that the redder regions are where the lichen is more abundant and the cooler areas are where they're less abundant. And then we can look at the difference between the two. And it says to us that it looks like this lichen has declined in parts of the drier boreal, and it's likely increasing a little bit in the forested regions. So those are our predictions that we can track over time. And when you add that together for all of the lichens and the mosses and the mites and the vascular plants and the birds and the mammals, you've pulled a nice cross section of biodiversity to try to understand how our coexisting species are changing as we change Alberta. One of the reasons why we really need to monitor multiple taxa is that it's kind of like doing a survey if you just went to your neighborhood and then said, well, this is what I think all of Edmonton feels like. You really want to have a broader cross section. And this is just a case in point where um, Dave Huggard, who's one of the great ecologists that works on ABMI, he looked at the different taxa and he looked at, how many of them were most abundant in different habitat types. So we have our sort of the five taxa across the top, your terrestrial taxa. And if it's in green, it's one of the top four. If it's in red, it's in one of the lower four categories. And we can see that lichens are really this conifer loving group. And conifer habitats are ones that are under perhaps some of the highest stress levels in the province. We know that the foothills is being impacted by all kinds of, competing demands, climate change by pine beetle and trying to manage the pine beetle. So we know that the lichens are vulnerable more than most other groups when we start to lose those conifer habitats. And lichens are also unique in that, as this is one of kind of maybe the assumptions people make, and in this case, it's true. Lichens do like older forests, so it just keeps getting better with age for these guys. And they have the least number of species of these five groups that actually likes young forests the most. So most of them tend to become more abundant and more common in older forests. And again, those forests are going to be the ones that are vulnerable as we go through things like climate change, greater insect and defoliation events, shortened fire cycles. So we can look at these lichens, we can start to understand them. But we have to remember that we're not monitoring lichens only for lichens' sake. We're monitoring them because we're using them as a surrogate. They're standing up for biodiversity in other groups that are more difficult to monitor. That's the assumption when you pick these surrogate taxa. And so we're starting to also look at lichens in different parts of the province that maybe, didn't traditionally consider lichens to be useful indicators, like the grasslands, and seeing how they might fit into something like a biodiversity management framework, which tends to be driven a little bit more by birds and vascular plants right now. So the province is working on these land use management frameworks and biodiversity management frameworks, and the ABMI data could be rolled into that. And some lichens like this, I think this is just a lovely lichen. It's called a yellow tongue lichen, and it's nice and big on your screen, but it's probably only about a centimeter tall in real life. But they're abundant and they grow under the grasses and on the selaginella in the grasslands of the province. Some of the other cool things we can do with these lichens is we can ask them more questions. So questions beyond just physical footprint. And, we talked about lichens being kind of infamous for telling us something about contaminants, dust and precipitation-borne contaminants. So we started a project a few years ago, um, basically trying to ask whether we could take these collections, these, hundreds of thousands of specimens of mosses and lichens that we're storing and keeping for research purposes and for future purposes that we maybe not even understand today. Could we look at them and ask questions about the contaminants right across the province? So we're doing that by working with the Wood Buffalo Environmental Association and some of their collaborators like Matthew Landis. And of course, they have a gold standard method of collecting lichens for specifically for contaminants monitoring, not for kind of broad scale biodiversity monitoring. And we took sites that were close to their research sites or within the area that they had done some good modeling for, and we extracted them and we looked at them with a few different kind of fancy techniques we don't need to go into. And we're going to compare that to the WBIA data and kind of use that as a check to see whether our samples can tell us something um, robust about the contaminant levels across the province. So where to from here? Well, sort of on a corny level, I'm gonna start by saying I think we should be like lichen. Lichens are resilient, lichens are tough, but they're also sensitive. They're community minded. There's no other organism I think that is more community minded perhaps than lichens. Um, and as Sheldon said, if we had to choose an organism to merge with, it really should be lichen. And he just gives a beautiful little analogy of there. So as we begin to be able to go to concerts again, I hope this will inspire you to dream of fusing yourself with a lichen and snacking on sunlight. Okay, so if that's not in the cards for us, at least not, in the next 10 years or so, then just start by going for a walk. There is amazing lichen diversity as we've discovered through the pandemic and being hunkered down in our own neighborhoods. We've documented over 110 species in Edmonton, and we're preparing a flora for anybody who wants to start to look at and learn about lichens in their urban outdoor classroom. Post images if you want to put names to things so that you can actually understand a little bit more about the organism. Take photos and put them up on Nature Links or iNaturalist. I would suggest though that make sure you zoom in. These guys are best appreciated at a scale that we're not quite used to thinking at. So you want to zoom in and take multiple pictures to help people out so they can help you out. You can take that beyond that and start to learn the boulevard beauties right outside your front door. So we've discovered that in Edmonton, there's an average of 6 lichen species on each tree surveyed. So that's not an overwhelming amount to start with if you want to learn a few. But up to 19 species can be found on a single tree in the right place. So you just never know what you'll find. You can check out some Lichen Art. Amanda Schutz did a project with us looking at the living valley or the lichens that make their homes in Edmonton. And there's lots of free resources, so we won't spend time on here, but if you're interested, I can share these resources with you after. And if you're you've done that and you want to take the next step, or if you're an academic scientist or somebody in industry and you want to kind of start asking questions with lichen, then we always welcome collaboration. We're having a lot of fun with you know researchers at different universities across Canada and the United States, looking at everything from how reindeer lichen grow, the things that caribou eat, to how grassland biocrusts are responding to grazing and drought, to what might actually be driving the different lichen communities and their ability to exist in different ecosystems of Alberta. So if you want to collaborate, then we welcome it. Whether it's ecology, taxonomy, there's so much to learn in lichens and so much to appreciate, whether you want to explore little tiny treasures or big, beautiful lichens like this Xanthoparmelia or shield lichen from the grassland. Because lichens really have a lot of stories to tell. And I think we've been compiling the words into the books, and now we need to begin to listen and to read and to understand. And I'm really looking forward to the next year where we have a lot more analyses planned to work with this priceless data set. So I'm going to hopefully leave you with your whys answered, and I'm going to acknowledge the team that makes this data set possible. Darcy and Murray have worked with me for over 10 years now. and Ermias Azaria, who's our senior ecological scientist who does the habitat modeling that we looked at today. And of course, the technicians, the cohorts of technicians, the hundreds of technicians that have collected these data and taken their work seriously in preserving knowledge of our natural diversity for generations to come. Thank you very much. And thank you to our sponsors and our partners.